next session is going to be led by Dr. Lund and Dr. Eichler. It is the Ask an Expert gene therapy. So do we have the doctors in the house? Yes, excellent.
Great, and in order to have this this panel, we invited two very talented people from industry to come and speak. This is Jacob Seeker, who is medical director at Bluebird Bio. Where's Jacob? And Eileen Sawyer, SVP of Clinical and Medical Affairs at Swan Bio. And I think this will be a really welcome opportunity to get an overview of different gene therapy approaches, and we hope then to have a nice discussion together with Dr. Lund and myself. So maybe since ex vivo gene therapy was the first to enter the realm of ALD, I asked Jacob to kick it off, and then we can go to Eileen.
Thank you, Florian. And thank you to ALD Connect for the invitation. With the next slide, I just want to make sure to state clearly that I'm a full-time employee of Bluebird Bio, and therefore I do have a financial interest. And also the presentation today and my answers to questions are not intended to be medical advice. I have two slides today that are really intended to just level set us for the discussion, but I'm very happy to answer the details in the Q&A part. This slide is intended to show where autologous hematopoietic stem cell-based gene therapy might fit into the spectrum of how ALD may manifest, and particularly for patients with cerebral ALD that are otherwise eligible to receive allogeneic stem cell transplantation. We hope that this type of gene therapy could be an option to use the patient's own cells instead of those from a donor. The next slide is intended to show what the treatment process entails for this type of gene therapy. It starts with mobilization using medication to move stem cells from the bone marrow into the peripheral blood, where they can be collected with a process called apheresis. The cells are then shipped to a manufacturing site. And for this type of gene therapy, it's important that the gene is added in a way that it integrates into the genome, because the blood stem cells are intended to replicate, form daughter cells, and it's important that it's a stable integration so that the gene remains in there. At the end of the manufacturing process, the cells are frozen and a battery of tests are performed to make sure that the product meets the specifications. And if the product meets specifications, then it's released, shipped to the treatment center, and once it's released there, the patient can come and can be prepared for the infusion. This preparation entails full myeloablative and lymphodepleting conditioning, similar to an allogeneic stem cell transplantation. It's essentially meant to make space in the bone marrow for the cells to engraft. There has to be some time for those conditioning agents to leave the body again before infusion of the drug product. And the hospital stay typically goes from a few days before conditioning until after the neutrophils have engrafted. These are the slides that I wanted to show to introduce to the topic. Really look forward to the discussion today.
I also have just a couple of slides to show. So I also have a very similar disclaimer. I'm a full-time employee of Swan Biotherapeutics, and again, this is not, anything I say is not medical advice. So just very briefly, for those of you who may not know Swan Bio or have been here yesterday as well, we're a very small gene therapy company. We were founded in 2018 by a trio of experts from different fields, some of whom are in this very room here today. And we're focused on bringing AAV-based gene therapy to patients who have devastating neurological disorders. And our first focus is adrenomyeloneuropathy. Go to the next slide. So AAV, or adeno-associated viral vector-based gene therapy, is a different process than what my colleague here was just describing. So AAVs are small, naturally occurring non-pathogenic viruses that we basically use pieces of to then deliver a helpful gene to the patient's cells. And this can be done directly to the patient as a one-time infusion. In this case, in our case, we'll be targeting the spine, so it's an infusion into the space around the cerebral spinal fluid, into the area around the spine that's filled with cerebral spinal fluid. And the AAVs are particularly well suited for adrenomyeloneuropathy because they're targeting the spinal tissue, which doesn't divide frequently, so you don't need to have integration into the cell's own genome, and AAVs don't do this. So they just deliver the helpful gene, and it remains there in the cell for the cell to use for the duration of its lifetime, or somewhere in the span of years onward. So we're working on, as I said, an investigational gene therapy that we hope to begin clinical trials with very soon. That's an AAV-based gene therapy. The two pieces that are sort of the lingo that's important to know about AAV gene therapy is the capsid, which is this outer shell. You can think of it kind of like the cardboard box that your Amazon delivery comes in. And the cargo, which is sometimes called the gene cassette. And that contains the piece that's going to stay in the cells and help them direct the cells to make the missing protein, in this case the ABCD1 gene. So that gets delivered in, it finds its way to your cells unwrap that cardboard box, they throw it out, and that gene cassette stays. And so, as I said, we're getting close to starting our first clinical trial. We hope to be able to share an update on that in the coming months. And I'll stop there.
Great. So I'm so grateful to both Jacob and Aline to speak, and also keep it so brief so that we have more time for questions to be asked. And because I think one thing we've noticed is that it's much better to have that engagement. And so maybe I have some questions here that came in online, but maybe we can start with the audience and see what's out there. Katie?
So I've spoken to Dr. Lund about this at length, and our Dr. Gupta at Minnesota as well. But my question is for the two of you regarding gene therapy. Is there any way to predict? My children have a unique mutation where they have a total deletion of the ABCD1 gene, and my son did go through the Bluebird Bio trial, my oldest son. But is there any way to predict whether or not an immune response would occur, like with what happened with Cruz?
Sorry, I don't. Cruz is
Cruz is my son.
my son.
Prediction of immune response, I would say there's no way to predict immune response given what we know about ALDP. Though if one has a full gene deletion of any gene, I think that would be a good predictor to be looking for an immune response. That's how I'd phrase it.
So I don't know if that's satisfying, and whether I understood the context. So you're saying immune response in the case of having received Bluebird Bio gene therapy, okay, and so I think. These are difficult questions to predict and have answers around. I think what was nicely described by Jacob is how you get hematopoietic reconstitution after the cells are delivered that now carry the lentiviral corrected gene. You know, if you so want, you're now have a chimeric situation where some cells carry the protein, others don't. It's an intracellular protein, and within that cell, it's on an organelle, so it's a subcompartment. So that might help a little bit, saying maybe it's not as exposed as you think of when you're thinking about extracellular aspects, but it's not something that we've come across or have thought or seen in the past. So, but always, yes, some caution. In general, I think it's very difficult for us to predict immune responses. Maybe we think of this in the context of some enzymatic defects that are complete nulls, but I think in ALD, we're not really in that situation of a metabolic disease where complete absence of something has ever led to us seeing immune responses. Fair enough, Troy? I don't know what you're... he's always a devil's advocate, so here we go.
Well, no. So all your proteins are actually on the cell surface because they're all presented in the context of HLA. It's just a little tiny bit of the protein. So your body's always sampling your cells, trying to figure out, are you foreign, are you friend, are you foe? So all your proteins do this. In other gene therapy trials, there are antibodies have been noted. In the MLD gene therapy trial, there are some patients that got antibodies to ARSA. In the Hurler syndrome gene therapy trial, there's some low-level antibodies to iduronidase. They don't seem to have an effect on the enzyme, though. In ALD, we've not seen anybody else generate antibodies to ALDP. We've looked, haven't seen it. In the case of a whole gene deletion, it's a very unusual situation that the baby, the child, has never seen the protein, ever. And so there's no sampling of friend or foe. So when you put the cells in, there might be a reaction like, oh, you look like foe, and you might have an immunologic reaction. That would be the only situation where we would think about it. But what the cause and effect would be from that is really unknown and more speculative.
So a few questions for the Swan Bio team. So I went on clinicaltrials.gov to look at the your first human study, and I think it's 20 patients or a little bit less, and there is a placebo arm. So my first question is, how do you do a placebo arm with gene therapy? Because when you give a small molecule, you can give a pill of sugar. When you're injecting gene therapy in the spine, how do you do a placebo arm? So first question. Number two is around toxicity. So recently there's been a lot of news around adverse events from gene therapies for pediatric genetic conditions. Many studies have had to stop or really go back and really rethink the study. So I would like to understand, what is kind of the threshold of toxicity and adverse events that you would feel comfortable with? So second question. And third question is efficacy, obviously a key metric in a first human study is to get some sense of baseline efficacy. And are you looking at, stopping progression of the disease or reversing some of the loss, knowing that, it's hard to regrow neural cells, but, would love your input on that.
Well, thank you. And so they cover a lot of ground there. So if I forget to address something in my response, please just remind me of the question. So I'm going to take them a little bit out of order. So first of all, let's talk a little bit about safety and first-in-human phase 1 studies. So this investigational gene therapy has been never tried in a human before. So the first and most important thing that we hope to learn from this trial is exactly around your second question there. What are the issues we need to be watching out for? Is it safe? That's question number one, well before we even get anywhere close to thinking about whether or not it has any benefits on the disease. And we are very much aware of all of the other activities in the field and what's been going on. And so as part of the trial, have built in some safety measures. Patients will be receiving immunosuppression because a lot of these adverse events and toxicities that you're describing do arise from activation of the immune system to some component of the gene therapy. And there's sort of multiple different branches of the immune system that can get activated in response to one of these therapies. And there are different medications that can be used to help damp down those responses. We'll be monitoring those patients very, very closely in the days immediately following the gene therapy, because that's when it's been observed the most of these responses occur. And we'll also be working very closely with external experts who will help us evaluate that data too. Certainly we hope that we will not see anything that suggests that this is too dangerous to continue, but if we do see that, we will need to stop the trial. Your second question was around blinding and efficacy. So those two are kind of related. So it is very difficult to blind a gene therapy trial, particularly one such as this that involves sort of very complicated, involves other medications. But essentially what we will do is we will do what's called a sham procedure or an imitation procedure. So only very few people will know whether or not the patient received the actual gene therapy. The patient will undergo pretty much almost everything exactly as if they're receiving the gene therapy, but they will not actually have anything infused into the spine. But they'll sit in the procedure room with the tubing on their back for the same duration as if they were having the infusion. And the hope is that even the patient themselves should not know or be able to figure out whether or not they received the active drug or not. Now, why do we have placebos in trials like this? That's a really important question as well. It helps us better assess both the safety and the efficacy of the drug to have a placebo. And in a case like this, where a lot of the assessments are either subjective or may have an effort-dependent component, we need to account for any sort of possible bias, whether it's a known conscious bias or an unconscious bias on the part of the patient or the person who's rating them on the test in assessing that. And so having a placebo and blinding the trial helps us remove some of that bias from the assessment, which is basically noise, and helps us hopefully get a better read on what's really going on a little bit faster. So bringing to the last question around efficacy, as I said, this is a phase 1/2 trial, and the first question is safety. But because this is a gene therapy, we also want to make sure that anytime we give someone a gene therapy, that there's also maybe a bit of a chance of efficacy. So we will be assessing efficacy in all the patients throughout the trial. And as I mentioned, we are blinding parts of the trial to help with that assessment. What we're really looking for first and foremost is halting of the disease progression. Of course, there is always the hope that in a disease like AMN, there may be the possibility to restore some function, allow some recovery of neurons that perhaps were sick but not gone. But I think as a starting point, what we're really looking for is just whether or not that progression can be halted. So hopefully I covered all of the ground that you covered, but great.
So with the way the AAV vector works, where it's, I guess, eliminated or after the cells die, it's no longer in the system after a couple of years, is this something you would envision as a multi-time treatment, or are you looking at it as a one-time gene therapy?
So that's a great question, and I'll comment and then maybe ask others to chime in as well. So at this point in time, we actually don't know how long an AAV gene therapy should last, whether or not that's in a disease like AMN or from what we've seen in other indications, other therapies using the same type of technology. So it's an open question. There are patients who have received AAV-based gene therapies that are years out now, but it's different target tissues. It may have to do with the cell types that are transduced. But there are some limitations right now that make it difficult to give another dose of an AAV-based gene therapy. And at the moment, we don't have the tools to overcome them, although there are many people working on them. So that's part of why we think of this right now as a one-time treatment.
Yeah, maybe just to briefly add to, there's the drug product or the vector as part of the platform, and then there's the disease biology part. I think in the past, when first AVs have been administered, and the little we know about the duration of effect has probably more to do with the state of the cells into which this is delivered into. And so in eye disease, there was clearly already some cell death occurring, in which case you lose drug product. And so just to remember that here in AMN, we don't think of the target cells, whether it's the neurons within the dorsal root ganglia, as actually dead. They're viable cells. So our hope is that there is long-term sustenance of that. But we will learn over time.
I had a question for Dr. Sawyer. Maybe a concern, not the concern, but more, I think, clarification that maybe the audience should know is, can you elaborate on, given that ALD is a familiar disorder and you may have multiple siblings in the same family affected by the disease, can you elaborate on the potential of antibody formation with AAV9 in siblings of treated patients who may then no longer qualify for AAV9 if they live in the same household? If you have ALD and I'm your brother and you get gene therapy, I might build antibodies since you bring the AAV9 home, and so the concern would be: is there a chance that then I would no longer qualify because I have built antibodies against AAV9? That's been raised in the muscular dystrophy world a lot, this issue. So they always make sure that it's mentioned in the consent form, it's clarified. So can you just elaborate on that a little? It is a real fact. It's happening in Duchenne.
So that's a good question. I was instructed I need to hold the microphone closer, so hopefully everybody can hear. I'm doing a better job on that. So yes, as part of that unpackaging and clearing out of the AAV capsid, it can end up in some of the sort of the bodily secretions. So I'm assuming that that's sort of where the question is coming at. So what happens? There's the chance for potential exposure to someone else in the household. So first of all, anything that is in those secretions should not be active. But these are questions that get looked at in the trials. And so that is something that's good to keep in mind. And I'm sure that there will be additional data that might help address that possibility.
Sorry, Ali, despite knowing you for a long time, I sometimes have some trouble catching up, but that's just me being slow. So in muscular dystrophy, the scenario might also be a little different, where you're talking about intravenous administration of AAV. So we do have the opportunity here of going intrathecally into a compartment we think of as relatively immune privileged and not interacting with blood to the same degree. So that might also be a significant difference. And yes, and there's clear screening happening as well.
We have some Zoom questions. Do we want to get to those? Or do we have another in person? Sophia.
Just to understand, so you said that for the Swan Bio, that men to help would go through chemotherapy. Would the men on the placebo also go through chemotherapy?
Sorry, to clarify, the AAV-based gene therapy does not require chemotherapy, but they may receive immunosuppression, so steroids and other drugs, but it's not the same as chemotherapy.
Okay. Would the men on the placebo also go on that though?
In order to maintain blinding, they need to.
Thanks.
Do we want to go to the online questions now?
So for Bluebird Bio, are patients with donor match eligible or only those with poor donor match?
So under the indication that we have been granted the accelerated approval for, there's no distinction between the available donor matches. But we have previously presented that we thought it's particularly an important option for those who have poor donor matches.
Okay, next Zoom question for everybody. Has neurological symptom progression been observed after gene therapy treatment if less score doesn't increase?
Yes, it has been observed. There is an association, though, with an increase in less score from baseline, which makes progression more likely to occur in patients who have less score increase. The way we have tracked neurologic function did not necessarily differentiate entirely whether this was due to CALD or any other neurologic cause. So we have observed, for example, an adverse event of transverse myelitis that has also led to neurologic symptoms, and we have incorporated those as neurologic symptoms as well.
Yeah. Just to maybe, I hear some assumptions behind that question. That if you have increases in less scores, that's there's clear correlation with symptom progression. We actually see after both bone marrow transplantation as well as after ex vivo gene therapy, as Jacob mentioned, increases in less scores, albeit not in that acute inflammatory fashion as in the natural history. These tend to be less associated with new symptoms arising.
In a similar, maybe related question, is lesion growth seen after gene therapy treatment, and does this correlate to symptom progression?
Yeah, I think that it's very similar, but definitely you see lesion growth after any type of transplant. So we always warn patients that your less score is going to go up, usually a point or two within the first couple of years, and that would be due to lesion growth no matter what kind of transplant you got. And the link to symptom progression is pretty interesting. The only way I can really try to answer is that we once graphed the neurologic function scores of all the patients we had seen over a few years. It was like 100 patients. And unless you were a less score of 9.5 or higher, they're almost all zero. So that's the kind of score range when we think the symptoms start. It's right around 9.5 or so. And interestingly enough, you've heard me say that a less score 10 is the higher risk category. It wasn't by accident that that's the higher risk category, because that's really where you're getting some symptoms starting.
Hi, for the Swan Bio group, you touched on this a little bit with the ability to take multiple doses. My understanding is that once you receive a dose of that, you have an immune response that would make you ineligible for another one?
Yeah, so that's correct based on sort of where things stand today. When you receive an AAV-based gene therapy, your body will produce antibodies to that outer packaging, that capsid. And so that will, at the moment, likely make it difficult for you to receive another type of AAV-based gene therapy, because they'll recognize that specific capsid, but they're probably also going to recognize other similar capsids.
And then, sorry, kind of related to that, also on Swan, how are you all going to measure success, and how long of a period will that be? And then obviously the treatment is targeted for AMN. Is it thought that that will also halt the cerebral development or future cerebral development?
Great questions. So as I mentioned earlier, the first objective of this trial that we're going to begin is safety. And so making sure that it's safe, well tolerated, as we understand the risks and how to manage them. Second, we are going to look at efficacy and do that over a 2-year time span. And here we're benefiting from a lot of the research that's already been done in this community and that you've heard about over the last couple of days, looking at what happens in patients who have AMN who are not treated. And we're also looking at that ourselves in a separate study so that we can really match up the assessments that we might do to look at efficacy in patients who have not received the gene therapy and patients who have. And that'll be a combination of measures like the 6-minute walk test that you've heard about, as well as some of the newer ones like balance or in wearables.
This is a related question. Is Swan Bio planning to do AAV for childhood cerebral ALD?
So our first focus is on AMN, and we are delivering it to the spinal cord directly. And so that's really the first step, and what we really think is most likely to be affected and benefit is the spinal cord disease from that approach.
So this is all fantastic news for people in the US. Do you ever foresee these treatments being available in the UK? The burning question from our families.
I'll start and then hand off. So we are starting the first phase 1 trial in the US. We do hope to move that to Europe as the program progresses.
Great news.
Yeah, for us, it's the situation that we can only offer the treatment at qualified treatment centers in the US. And patients that are coming from other countries would need to be treated in the US.
It's just, so one of our families asked me this question that Libmeldy for MLD has been passed by the NHS as a treatment for certain types of metachromatic leukodystrophy. And obviously you were starting to contact the NHS because we were very involved in that process. And I do understand that, it's very difficult, so I'm not accusing anyone of pulling out. It's just that obviously we have lots of families who wouldn't necessarily be able to afford to come to the States to be treated.
Yeah, I can share we all see the same issues, but it has been quite recent that we needed to make the decision to focus in on the US. And that, for the moment, remains the focus and the option that we have available.
Any other questions out there? Nothing online? All right, let's give a round of applause to our very brave panelists and moderators. Thank you guys very much.